My name is Joshua Gabi. I welcome you to Inheritance and Evolution, Topic 7, our learning outcome, structure and functional significance of nucleic acid in meiosis and mitosis, their role in cellular function, and how mutation in nucleotide sequences can contribute to disease like cancer. Our learning activity today is coming from D in group learners search and discuss mitosis and meiosis, cancer causes, risk factors, prevention, and management. Then they make their write-up and include the mind map showing the relationship between cell division and cancer. So we are starting with mitosis and meiosis. We shall look at the growth of a baby. A mother brings her 2-year-old child to a clinic. The doctor explains that the child has gained height because cells in the bones and muscles have been dividing rapidly. Task: which type of cell division causes growth? Explain why this type of division is suitable for growth. State the chromosome number change during this process. Which type of cell division causes growth? The type of cell division is mitosis. Explain why this type of division is suitable for growth. Why suitable? It produces genetically identical cells. It maintains the same chromosome number, ensures tissue grow and repair consistently. State the chromosome number change during this process. The chromosome number remains diploid, 2n to 2n. Now, still under mitosis and meiosis, we want to look at the production of gametes. In the school laboratory, students examine slides from tests and ovaries. They note that these cells divide differently compared to the body cells. Each resulting cell has half the normal chromosome number. Task: identify this type of cell division. Explain why the chromosome number is halved. Describe how meiosis increases genetic variation. Identify this type of cell division. The type of division is meiosis. Explain why the chromosome number is halved. Why halved? To ensure fertilization restores diploid number, prevents chromosome doubling in each generation. Describe how meiosis increases genetic variation. Genetic variation contributions, crossing over, that is the exchange of genetic material, is going to bring about genetic variation, independent assortment of chromosomes, and random fertilization. Now let us look at a cell with an ELA, still under mitosis and meiosis. Item 3. A student views a cell in which chromosomes do not separate properly during meiosis. Some gametes receive extra chromosome, others receive fewer. Task: name this meiotic ELA. Give an example of condition caused by this ELA. Explain why such ELA are rare in mitosis. Name the meiotic error. The error name is non-disjunction. Give example of a condition caused by this error. Condition examples we have Down syndrome, trisomy 21, Turner's syndrome, XO, Klinefelter syndrome, XXY. Explain why such errors are rare in mitosis. Why rare in mitosis? Mitotic spindle checkpoint tightly regulate chromosome separation. Errors lead to cell death rather than survival. Let's now talk about cancer, causes, risks, factors, prevention, and management. We are going to look at a family with a history of breast cancer. Item 4. A woman visits a hospital because several relatives have developed breast cancer at young ages. Task: identify one genetic risk factor for cancer. Explain why family history increases cancer risks. Suggest screening methods for early detection. Identify one genetic risk factor for cancer. Risk factor BRCA1 or BRCA2 gene mutations. Explain why family history increases cancer risks. Explanation: inherited mutations increases the chance of cells becoming cancerous. Suggest screening methods for early detection. Screening can be carried out using mammography, genetic testing, and clinical breast examinations. Still under cancer, causes, risk factors, prevention and management, we want to look at workplace exposures. Item 5. A factory worker is exposed daily to industrial chemicals and cigarette smoke. Task: state 2 environmental causes of cancer shown in this scenario. Explain how carcinogens cause cancer. Suggest preventive measures. State 2 environmental causes of cancer shown in this scenario. Environmental causes, we have the cigarette smoke, chemical carcinogens like benzene, asbestos. Explain how carcinogen cause cancer. How carcinogens cause cancer: they damage DNA, causing mutations, leading to uncontrolled cell division. Suggest preventive measures. Prevention can use protective equipment, reduce exposure, workplace safety monitoring, and stop smoking. Cancer causes, risk factors, prevention, and management. We want to look at skin cancer in hot climate. Item 6. A young adult develops suspicious dark spots on the skin after years of working outside without sunscreen. Task: what is the likely cause of this cancer? Explain why this cause is dangerous. Suggest 3 prevention methods. What is the likely cause of this cancer? The likely cause is ultraviolet radiation from the sun. Explain why this cause is dangerous. Why dangerous? Ultraviolet light causes DNA mutations in skin cells. Suggest 3 prevention methods. Prevention: use of sunscreens, wear protective clothes, limit direct sun exposure from 10 a.m. to 4 p.m., regular skin checkups. Still under cancer causes, risk factors, prevention, and management. Let's look at the management of a cancer patient. Item 7. A patient diagnosed with colon cancer begins therapy, including surgery and chemotherapy. Task: state 2 treatment options used. Explain how chemotherapy works. Give 1 side effect of chemotherapy and why it happens. State 2 treatment options used. Treatment option includes surgery, chemotherapy, radiotherapy also possible. Explain how chemotherapy works. How chemotherapy works. It stops or slows rapidly dividing cells. Give one side effect of chemotherapy and why it happens. Side effect. There is hair loss, nausea, immune suppression. All this because chemotherapy affects all rapidly dividing body cells, not just cancer cells. Relationship between cell division and cancer. Want a mind map. We have normal cell division and abnormal cell division, which results into cancer. In normal cell division, the cell division is regulated. In abnormal cell division is uncontrolled. In cell division, they are controlled by checkpoints. Abnormal cell division mutation is proto-oncogenes. Normal cell division produces healthy cell. Abnormal cell divisions become oncogenes. Normal cell division occurs via mitosis and meiosis. Abnormal cell division occurs in mutations in tumor and suppressor genes. Cancer results when normal controls of cell division fail due to DNA mutations, leading to uncontrolled mitosis, resulting into tumor formation. This can cause a possible spread of cancer to all parts of the body. Metastasis. Causes of cancer. Carcinogens, radiation, viruses like HPV, inherited mutations, lifestyle, smoking, alcohol, diet. Effects: tumor formation, organ failure, pain, bleeding, and death. Outcomes: cancer cells spread, metastasis, fatal if untreated. Thank you very much for participating in inheritance and evolution, our learning outcome, structure and function significance of nucleic acid in meiosis, mitosis, their role in cellular function, and how mutation in nucleotide sequence can contribute to disease like cancer. We have been looking at learning activity D, where learners search and discuss mitosis and meiosis, cancer causes, risk factors, prevention, and management. Then they make up their writing, including the mind map, showing the relationship between cell division and cancer. If this presentation has been very useful to you, visit me at Mugabe Joshua Biology on YouTube.